We have a lot of time, plenty of time for questions and really fantastic talks. I don't know if we can turn the lights up a little bit too. So have a seat. I guess I'll start with a question. So David, you showed a picture of what pancreatic cancer cells looked like with FAK inhibition, and the architecture looked a little different. Do you notice any sort of disaggregation or loss of tertiary structure in the cancers, and with the loss of the stromal elements? That was just an observation of mine from one of your slides.
Yes, I can tell you what it looks like preclinically, but we haven't processed all the clinical samples we have to date. We're doing that in batch. Preclinically, we've taken advantage of a lineage tracing system developed by Ben Stanger and Andy Rim to sort of be able to analyze the extent of invasiveness. So you'd see a loss of the stromal density, but not a complete deletion or depilation. And what you find is there's less single cell invasion and less evidence of invasion and more glandular architecture in general.
Okay, thank you.
But not, remember that unlike in vitro, we never obtain a kinase inhibitor in vivo never obtains full inhibition of the target for the full period of time. So, this may be different from knockouts or in vitro studies.
Thanks. Shoop.
Yeah, my question was also for David. Thanks for that really exciting talk. I had two questions, actually. The first was, I think as you just alluded to, kinase inhibitors obviously have multiple targets, and this can be kind of a double-edged sword. So in the clinic, it can lead to off-target effects, reducing your dose exposure and your inhibition. But preclinically, it may also enhance your effect artificially by targeting other kinase pathways on which the tumor cells rely. And so I was wondering if there is like a conditional knockout that you could sort of test in your studies to show specifically that the tumor cell FAK genetically is actually required for tumor progression. And then the next question was about sort of how you came to decide to focus on FAK, and if there are other sort of tumor cell derived factors that you were thinking of looking at.
First question, it's a great question. So the off-target or the other targets of the inhibitors we use are PIK2, this is FAK2, which is actually a stromal FAK, and FAK and leukocytes. And FLT3 can be hit, though 10 to 20-fold lower. We tested FLT3 in the model. We tested FLT3, whether we were hitting that target looking at DC development. We weren't. But that said, your approach is exactly, in data I didn't show you, we shRNA, and we also tried CRISPR, our discussion this morning. We shRNA knocked out FAK1 in tumor cells. We can recapitulate the responses in the tumor microenvironment. That said, PIK2 is important in the tumor microenvironment, which suggesting that while this paper focuses on the tumor intrinsic effects of FAK1 and generating a pro-fibrotic, pro-inflammatory response, we're also looking at the tumor microenvironment effects. I just don't have all that data yet. It's a great question. Second question is, how did we get to FAK? So this is intelligent by design slide presentation. PIK2 is involved in monocyte trafficking, which is another interest in the lab. That's actually how we got the molecule and then figured it did different biology. So serendipity rather than intelligent design.
Other questions? Katie?
I have a question for Eric regarding potential surrogate biomarkers in advance of doing TMG analyses of TILs. Have you looked at any next-gen sequencing neoepitope prediction plus allele frequency algorithms to try to predict from your melanoma experience and your solid tumor experience which patients might have the most successful results from adoptive cell transfer?
Yeah, that's a good question. So as you're probably familiar with, at least immune checkpoint blockade, PD-1, CTLA-4, there's a correlation between mutation burden, mutation load, and clinical responses. Unfortunately for our cohort, for the gastrointestinal cancer cohort, we just don't have enough patients really and response rates to really draw any conclusions. For example, Melinda's case is 26 mutations. and she really is one of 2 responders. The other patient had 61 mutations, and she was another responder. So there are, at least for metastatic GI cancers, I don't think we can draw a correlation. For melanoma, I don't think that analysis has been run in terms of neoantigen load and clinical responses to adoptive cell therapy.
Or just the neoepitope algorithms. I know Foundation and a variety of places are working on trying to predict what will be antigenic structurally.
Right. Yeah, so those algorithms, you're speaking about the HLA peptide prediction binding algorithms. Again, in the setting of melanoma, we have not looked at that. We do know that when we predict neoantigens using those algorithms and softwares, that only a very small percentage of those will actually be true T-cell targets at the end when we test against TIL. So it's a very good question. I know trying to find biomarkers to stratify patients who will respond, but again, just based on our limited experience with patients who have responded in low mutation burden, and right now I don't think we can use that for adoptive cell therapy.
Eric, I have a follow-up question. I was struck, and you commented on the end of the overlap between a functional requirement for a gene and a neoantigen. And so the approaches that you talk about are largely looking for neoantigens, and the question would be about resistance. So are you thinking about a functional approach in terms of identifying proteins that we haven't yet recognized our requirements and then overlap with the neoantigen search?
I think that'd be a great idea, except that right now when we test even against a whole set of mutations from one given patient, we're only finding neoantigen reactive T cells maybe against 1 to 5 of those neoantigens. So we can't really be too choosy at this point.
Sure.
But ideally, yes, we'd like to target neoantigens that are functionally important for the cell. And I think I mentioned in my presentation that the other responder actually was a patient with colorectal cancer where we targeted mutated KRAS G12D, and she was a responder. The intriguing thing with that case is that, so she had 7 lung lesions. We gave her approximately 150 billion TIL, and 75% of those were reactive against mutated KRAS G12D. 6 out of the 7, all 7 lesions shrank, except for 1 lesion that actually progressed at 9 months. And so I know a lot of groups are now searching for immune evasion mechanisms to checkpoint inhibitors and other immunotherapies. This patient, we actually resected the progressing lesion, and we found that the tumor had lost haplotype of chromosome 6. And why is that important? Well, chromosome 6 encodes for your HLA molecules, which is required for T cells to recognize antigens. And so maybe it's not surprising, but the haplotype that was lost actually had the HLA that was required for the KRAS reactive T cells to actually recognize the tumor. So that's an immune evasion mechanism, even though we targeted a driver mutation, which is the ideal, I think, antigen to go after. Tumors can find alternative ways, and that's why I think targeting a broad repertoire of antigens might be a bit more safe, because when a tumor loses haplotype, it still has 3 HLA. And so if you can find T cells that recognize tumor antigens in the context of those HLA, then it might be more effective. Thank
you. Other questions? I'm gonna end with one before we all go dance next door. So Nabeel, I don't talk to you for a while, and then you come back with a whole new pathway, fully explored and understood on 7 different levels. So what's the next sort of gene or rare subtype you're going to figure out? Do we get a preview for next year?
My guys in my lab will just bring me something, and I'll talk about it.
If they want to have jobs in Rochester, I'm happy to talk to them. So we're ahead of schedule. Any other questions? Yeah? Okay.
Mario Bosco, Yale University. I found the talk about cyclic MP really fascinating. Do you have any idea which kind of adenine cyclases is being activated, and also the intracellular localization of the PKA that is relevant for tumor formation?
Yeah, so that's a fantastic question. So I think for most of the audience, the issue is that one of the ways that you can get this really dichotomous function of cyclic AMP PKA signaling is that PKA is anchored in different parts of the cell, as well as enzymes that degrade cyclic AMP. And so the complement of those and the localization, and that's something that we're extremely interested in, and for the context of this cancer type, but also some other cancer types where these lesions happen. But we don't know yet, but we're very, very interested in that.
If I can ask a second question. So there's a connection between, and you mentioned that cyclic MP, PKA, RAS, RAF, ERK. That would predict an effect of RAF inhibitors, for example.
So
Sorafenib, just to check.
yes. So here, one of the ways that the signaling pathway has these yin-yang effects is by, in certain cells, cyclic AMP signaling is a very strong inhibitor of the ERK pathway. Turns out in these cells that's the case. So it's not
an inhibitor.
Yeah. So it's not increasing. It's not increasing ERK signaling. It goes down somewhat. So it's using a completely different set of pathways. Certainly, it's still active. RAS is these tumors require RAS, no question. But it's not acting as an amplifier of ERK signaling. And Richard Maris worked on this quite a bit a number of years ago in many groups. We think that this biology is very different. It doesn't involve sort of effects on canonical RAS signaling as being pro-tumorigenic.
Thank you.
So thank you. We have a break till 10:30. Just a note on the sheet.